Assalamu alaikum class. Today our topic of discussion is the integumentary processes in the insects. Insects integument bear a great number of different processes. Their integument is the externalmost structure in the insect body, and there are two types of the insect integumentary system. First one is the non-cellular. These processes are entirely composed of cuticle. It's mean that no living cells are present in it, and may take the forms, many forms, for example spines, ridges, nodules, and the microtricha. Second one is the cellular. It's mean here living cells are present, or you can say all the conditions of living cells are complete. Cellular processes are of two types: unicellular processes. These all are referred to as the CT, but they may be hair-like or flattened into the scales, may bear branches and appear plumose, or may take on other shapes. The cetal shaft is formed by the protoplasmic growth of the special trichogen cell. I will explain you this in the next diagram. Here you can see the diagram. This is the epicuticle. Actually, this is the cuticle layer. It is further divided into epicuticle, exocuticle, and the endocuticle. Now the epicuticle has various regions. Outermost is the cement layer, this one. Below the cement layer, wax layer is present. This is the outer epicuticle, and this is the inner epicuticle. This is the sensillum, or hair-like projection, that is projecting outward from the cuticle. Here now you can see the full diagram. This is a trichogen cell from which hair shaft originates, and this is the chromogen cell which forms the socket in which from which the hair shaft originates. These are the trichogen cells, or the hair cells. Now, what are the functions of the CT? If CT are present in then what is their function in the insect body? They act as a sensory structures, and they act as a secretory structure. It's mean they are involved in the perception of tactile stimulus, as well as they are involved in the release of some kind of secretions, which may be either hormones or any other kind of secretion. Multicellular processes, these are hollow outgrowths of the integument, and are lined with the epidermal cells. They generally take the form of the spines. Outgrowths of the cuticle, there are two types of cuticular outgrowth. First one is the cuticular appendages. These are outgrowths of the cuticle that are connected with the membranes. Okay, these outgrowths are connected with the membranes. No hard covering is present between them. Now there are further divisions of cuticular appendages, that are the spur multicellular appendages, and these are found in the legs of many insects. CT, hair-like structures produce singly hypodermal cell, which enlarged and called as the trichogen cell. It was previously seen in the diagram. Next is the membrane sporting CT is produced by another cell called the tormogen cell. Next is the type of CT. Clothing hairs. It covers the either general body surface. When these are provided with the thread-like branches, these are known as the pulmose hair. When they are stout and rigid, they are known as the bristles, and they are found in the fleas and the trichinida. If they cover the whole body, they are known as the clothing hairs. CT, they are called scale use, and these are present in the wing of the Lepidoptera. You are familiar with the butterflies and moths. Glandular CT, they have canals for the secretion of hypodermal glands. In the previous page, it was discussed that CT have sensory function as well as secretory function. So if they are secretory in nature, they are known as the glandular CT. Sensory CT, they are associated with the nerve cells and act as the tango receptors. This is the diagram. Here you can see the dermal glands. This is the epidermis, and this is the modification in the epidermis, which is known as the dermal gland, and this is the duct of the dermal gland, and it is the pore which open on the surface. This is exocuticle, this is endocuticle, and this is the trichogen cell, which is the shaft of the hair, and from which bristles are originated. Cuticular processes. These are cuticular outgrowths which are having a membranous joint. And now there are further deviants of the cuticular processes: microtricha, spines, special projection, and the integumental glands. Minute hair-like structures found in the these are present in the wings of the diptera, order Diptera. Spines, these are the hollow outgrowths of the cuticle which may be multicellular. Special projections, these are present like stridulating organs, stylet apparatus. Stridulating organs, these are involved in the production of specific sounds which are used to attract the mate. Integumental glands, modified hypodermally, which perform special functions, and saliva gland, silk gland, scent glands, these are included in the integumentary glands. Insects are well endowed with the cuticular extensions varying from fine and hair-like to the robust and spine-like. 4 basic types of protuberance can be recognized on morphological, functional, and developmental grounds. Spines, they are multicellular, made up of more than one cell with undifferentiated epidermal cells. Epidermal cells are not differentiated here. CT, also called as hairs, macrotracheae or tracheal sensilla, are multicellular with the specialized cells. Acanthi, they are unicellular in origin. Microtricha, they are the subcellular with the several to many extensions per cell. Here this is clear in the diagram. This is the epidermal layer, and here you can see various epidermal cells in the diagram. So this is the multicellular spine, and above the epidermis, cuticle is present. This is the CT on the trichoid syncytium. CT is evolving from the epidermis, and this is the trichogen cell, and this is the tormogen cell, which is the socket in which trichogen cell is present. Next is this is the diagram of acanthy. Epidermal cells are present in a single line, and protuberance are originated from its surfaces. This is the microtrichae. Here you can see now what is the basic difference between the microtrichae and acanthy. There यहाँ पे कम projections हैं सेल के ऊपर, मतलब पर सेल सिंगल प्रोजेक्शन है, लेकिन यहाँ पे पर सेल मोर देन 2 या उससे ज़्यादा प्रोजेक्शंस हैं माइक्रोट्राइके पे. CT are the sensory organs present in the insects, as well as bristles are also present. These are also involved in sensory detection. Here in this diagram you can see that detailed structure of cuticle. The outermost layer is the cement layer. Below the cement layer, wax layer is present, and this layer is known as the superficial layer. Outer epicuticle is present, inner epicuticle is present. This is the exocuticle, and various glands which releases the secretions on the surface of the cuticle, they are present here, and these are all the derivations of the epidermis. These are the pore canals, and these canals open onto the surface of the cuticle, and these are the wax filaments. That is the completion of the topic. Thank you.